My name is Gregor Mendel. I'm from Austria and I work in the monastery as a monk. I was born in 1822. I live and spend my time in my parents' barn until I turned 11 and a schoolmaster told me and my family that it was a good idea to go to the middle school. Though it was kind of expensive for us. We found a way and I graduated from my studies with honors in 1840. 3 years later, against the wishes of my father, who expected me to take over the family barn, I began studying to become a monk. I joined the Augustinian order at the St. Thomas Monastery in Brno, where they gave me the name Gregor. My original name was Johann. At that time, the monastery was a cultural center for the region, and I had access to the research and teaching of other monks, the monastery's extensive library, and experimental facilities. A few years later, I got extremely tired from the monastery and left to become a certified teacher, but because of one or another thing, I couldn't. I went back to a monastery in 1853, where I began my most famous experiments such as the transmission of hereditary traits in plant hybrids. I chose to use bees for my experiments due to their many distinct varieties, and because offsprings could be quickly and easily produced. In 1865, I presented the results of my experiments with nearly 30,000 pea plants to the local Natural History Society. Based on the patterns I observed, the counting data I collected, and a mathematical analysis of his results, I proposed a model of inheritance in which characteristics such as flower color, plant height, and seed shape were controlled by pairs of heritable factors that came in different versions. One version of a factor, the dominant form, could mask the presence of another version, the recessive form. The two pair factors separated during a gamete production, such that each gamete, sperm or egg, randomly received just one factor. The factors controlling different characteristics were inherited independently of one another.
My laws. The law of dominance. Dominance describes the relationship between two alleles. If an individual inherits two different alleles from each of its two parents, and the phenotype of only one allele is visible in the offspring, then the allele is said to be dominant. My law of dominance states that if one parent has two copies of allele A uppercase, the dominant allele, and the second parent has two copies of allele A lowercase, the recessive allele, then the offspring will inherit an A uppercase and A lowercase genotype, basically a heterozygous genotype, and displays a dominant phenotype. The law of segregation. My law of segregation states that for any trait, each parent pairing of genes split and one gene passes from each parent to an offspring. Which particular gene in a pair gets passed on it completely up to chance. The law of independent assortment. In my law of independent assortment, I explained that different pairs of alleles are passed on to the offspring independently of each other. This means inheritance of genes at one location in a genome does not influence in inheritance of genes at another location.
I didn't discover these foundational principles of inheritance by studying human beings, but rather by studying Pisum sativum or the common pea plant. My data did not support ideas about traits blending that were popular among the biologists of my time. Bees were a good model system because I could easily control their fertilization by transferring pollen with a small paintbrush. This pollen could come from the same flower, self-fertilization, or it could come from another plant's flower, cross-fertilization. First, I observed plant forms and their offsprings for 2 years as they self-fertilized, or self-fed, and ensured that their outward measurable characteristics remain constant in each generation. I had to determine what happens when two plants that are hybrid from one trait are crossed with each other. But I also wanted to determine what happens when two plants that are each hybrid for two traits are crossed. I therefore decided to examine the inheritance of two characteristics at once. The observations that I made and what I discovered were critically important because the leading theory in biology at the time was that inherited traits blend from generation to generation. Most of the leading scientists in the 19th century accept this blending theory. Charles Darwin proposed another equally wrong theory known as pangenesis. This held that hereditary particles in our body are affected by the things we do during our lifetime. These modified particles were thought to migrate via blood to the reproductive cells and subsequently could be inherited by the next generation.
As most of us know, generally through the history, almost every scientist is inspired by another scientist's work. In my case, my greatest influences were my professors. When I was 20 years old, Friedrich Franz advised me to continue studying science. So I followed his advice because of 1, I could get financial help, and 2, I realized that I was more interested in science than in religion. Other of my professors at university, Johann Carl Nestler, tried to explain the importance of careful breeding by noting that nature is capable of producing species of animals and plants through forces men, and that breeders control the reproductive process, such as inbreeding or outcrossing. What led me to write about the way in which breeders could control traits and why. After finishing my studies, I started teaching at Brunn Modern School, where I met Professor Alexander Makowski. After meeting him for a while, we became associates. He was interested in the study of flowers, which inspired me to investigate plants in order to determine hereditary likelihood. As a fact, I was inspired by Darwin's work because I didn't believe what he said about the variability and environmental change, which encouraged me to make my own research about the topic. One of the best ways to predict the outcome of genetic cross is by drawing a simple diagram known as Punnett square. Punnett squares are mathematical probability to help predict the genotype and phenotype combination of genetic crosses. Constructing a Punnett square is easy. You begin with a square. Then, following the principle of segregation, all possible combinations of the alleles in the gametes produced by one parent are grading along the top edge of the square. The other parent's alleles are then segregated along the left edge. Next, every possible genotype is grading into the boxes within the square, just as they might appear in F2 segregation.